So in this lab, we're going to look at a standard plate count, and this is activity 6-1. So the purpose of the standard plate count is to isolate colonies of bacteria from a large population and determine the titer of the stock culture. So an example of a stock culture is a liquid culture that is used for a streak plate. And when we refer to a titer, we're basically referring to the number of cells per milliliter. So what we're essentially trying to do is to quantitate how much bacteria is in that stock culture. Now, when you look at the purpose of the standard plate count, notice the definition or the overall purpose. Isolate colonies of bacteria from a large population. What does that part sound like? What other technique did you look at that we isolated colonies from a large population? And the answer is that is from a streak plate. A streak plate is used to isolate colonies from a large mixed culture. In this case, it's not a mixed culture, it's a pure sample, and we want to isolate individual colonies. And essentially what we're trying to do is determine the concentration of bacteria in that sample, meaning how many bacterial cells per milliliter are present. And so that's what makes this different than a streak plate. In a streak plate, remember that we're streaking for isolation, and that in our 4 quadrants, quadrant number 1 has the most bacteria, then quadrant 2, 3, and 4. And so if we were to look at a streak plate, is there a way to determine the concentration? And the answer is no. There's no way to determine the concentration when looking at a streak plate, because quadrant number 1 is going to have a lawn. It's going to have so much bacteria that you can't count it. So a streak plate is not going to allow you to quantitate the number of bacteria. So instead, we would do a standard plate count. We're going to isolate individual colonies, and once we do that, we will be able to quantitate or determine the titer of the stock culture. And so when we do this, this would be basically, the idea is that we first will perform what's called a serial dilution. And that is that we will dilute the cultures to different concentrations, and then we will plate the different concentrations and look for the plate that has isolated colonies. We can then quantitate the number of bacteria that's in that original culture. When we do this experiment, we will be using a safe strain of E. coli. We have E. coli in our gut. E. coli is part of your normal flora. Often when you think of E. coli, you probably think of like the E. coli that makes you sick, like outbreaks that like Chipotle had, for example. And that's not just standard E. coli. That's not the E. coli that's found in our gut. That is a particular strain of E. coli called O157H7. That strain of E. coli has a toxin that it produces that makes you sick. It produces what's called a Shiga toxin. And that toxin is what gives you, violent diarrhea and upset stomach. But standard E. coli we have in our gut, and it doesn't really cause any problems. And so in this experiment, we would be using a safe strain of E. coli, one that would not be pathogenic. A streak plate is not useful to determine the number of bacteria. And so that's what we just talked about. So we can't do a standard streak plate if we want to quantitate the amount of bacteria. Yes, a streak plate can be used for isolation, but we can't determine the titer using a streak plate. We have to use a standard plate count. When you would do this experiment in the lab, this would be done in pairs. You and your partner would do this experiment together. So we need to start out by talking about serial dilutions. So in a serial dilution, we take our stock culture and we're going to continually dilute that. We're going to dilute it more and more each time. We need to talk about how do we do calculations for our standard plate count? How do we determine the dilution that's in the tube? So what we're doing is we're taking our stock culture and we're transferring 0.1 milliliters into 9.9 milliliters of blank. What that is that's essentially sterile water. So when we calculate our dilution in that first tube, we need to take 0.1 milliliters divided by the total volume. The total volume is the 9.9 plus 0.1, so that's 10 milliliters. So my dilution is 0.1 milliliters over 10. Now, the easiest way to put this in an exponent is to make the top number 1. And what that means is that you're going to move the decimal point one place to the right, which means we have to do that on the bottom as well. So the decimal point would be here. It would move one place to the right. That little loop is going to get a 0. So we get 1 over 100, right? Because we moved the decimal place one place to the right on the top. We also moved it one place to the right on the bottom. And so that's why we get 1 over 100. Now, 1 over 100 is the same thing as saying 1 over 10 to the second. 1 over 10 to the second. So 100 is a 1 with 2 zeros. So 1 over 10 to the second. And that is the same as saying 10 to the negative 2. So 1 over 10 to the 2, you're going to make the exponent a negative number. So in this case, your dilution in this tube, 10 to the negative 2. That's your dilution in that tube. Now, that is your, what we call your final dilution factor. Final dilution factor is going to be the amount that that tube is diluted. Your dilution factor, meaning the amount you diluted it, is 10 to the negative 2. This is your dilution factor. That's how much you're diluting it each time. The tube, that first tube, has a final dilution factor of 10 to the negative 2. That's the dilution in that tube. Now, notice you're going to take from your 10 to the negative 2, and you're going to transfer 0.1 milliliters into 9.9 milliliters blank again. So notice that what you're doing is you're diluting this 10 to the negative 2 more, right? So you're diluting it 10 to the negative 2 more. But in this case, you're starting with a tube that is already diluted 10 to the negative 2. So what you need to do to determine the concentration of this tube is you're simply going to add the exponents. So if I start out with 10 to the negative 2 and I dilute it 10 to the negative 2 more, that means that my dilution in this tube is 10 to the negative 4. That's my final dilution in that tube. So now I have a tube that is diluted 10 to the negative 4, and I'm going to dilute it 10 to the negative 2 more. So I'm going to dilute it 10 to the negative 2 more, but I'm starting with a dilution that's 10 to the negative 4. So in that next tube, it's going to be 10 to the negative 6. So that is going to be your final dilution in that tube. So the difference between the dilution factor and the final dilution factor, the dilution factor is the amount that you're diluting each time. The final dilution factor is what is the dilution in that tube. And that takes into consideration the dilution that was before it. So this tube, the second tube here, that tube started with 10 to the negative 2. We diluted it 10 to the negative 2 more. So the final dilution is going to be 10 to the negative 4. That's the dilution in that tube because we didn't start with the stock. We started with a sample that was already diluted 10 to the negative 2 more. And so this is how we calculate our dilution factors. Now, when we do this serial dilution and we end up with 3 tubes, 10 to the negative 2, 10 to the negative 4, 10 to the negative 6, those are my serial dilutions. Those are what I start with. Then what I would do is I would transfer that onto a plate. Now, remember that I said that my titer is per milliliter, the number of bacterial cells per milliliter. So if I'm transferring from my 10 to the negative 4 and I'm transferring 1 milliliter, my dilution factor is still 10 to the negative 4. So that plate would have a final dilution factor. That's what's written on the plate. The final dilution factor would be 10 to the negative 4. Because I start with my tube that's 10 to the negative 4, I transfer 1 milliliter, and I plate that on a plate. And so my final dilution factor on that plate is 10 to the negative 4. Because again, my calculations are per milliliter. So if I transfer 1 milliliter, my concentration stays the same. However, if I start with my 10 to the negative 4 plate, and now I only transfer 0.1 milliliters, so because it's only transferring 0.1 is 10-fold less than 1, right? That would be moving the decimal point 1 place. And if I move my decimal point 1 place, then my exponent also needs to move 1 place. And because it is less concentrated, I'm not using 1, I'm using 0.1. That is using 10-fold less bacteria. So now my final dilution on my plate is going to be my 10 to the negative 5. I move the exponent 1 place. Now it goes negative 5 rather than negative 3 because negative 5 means that it's more dilute, right? We're using 10-fold less bacteria. Then we have our 10 to the negative 6 plate, and we transfer 1 milliliter. So notice that our next plate is going to be our 10 to the negative 6. The dilution stays the same. And then lastly, we have our 10 to the negative 6, and we transfer 0.1, which our last plate then would be 10-fold less, 10 to the negative 7. And so the final dilution factors are what are written on the plates. We're going to write our final dilution factor on our plate because that is the final dilution that's on that plate. Now, one of the things to note, and I'll describe this again as we go through this experiment, is the volume that's in these nutrient agar deeps. What that means is basically that's agar that is still liquid. And what that means is, remember when we make agar, that the agar is going to liquefy at 100 degrees Celsius and it will solidify at about 40 degrees Celsius. So when we're doing this experiment, these plates are actually empty when we start, meaning they don't have any agar in them. What we do is we take the agar that would normally be poured in a plate and we use that agar and we actually mix the bacteria with it and then pour it on the plate and let it solidify into a plate. So oftentimes when students are trying to calculate the final dilution, they're worried about the amount of volume that's in the agar deep. That doesn't matter. Don't use that as your calculation for your final dilution factor because it doesn't matter how much agar is in that tube. What matters is how much bacteria is being transferred, because that's the concentration you're trying to determine. So that's why if you take your 10 to the negative 4 and you transfer 1 milliliter, you're going to end up with 10 to the negative 4 as your final dilution factor. Even if the nutrient agar deep has 30 milliliters or 40 milliliters, that won't matter. The concentration is dependent on the number of bacteria in the stock culture. So what I need to show you next is I need to show you how do we use what we call a micropipettor. And a micropipettor is used to transfer small volumes because notice we're going to transfer 0.1 milliliters. So we need to look at and see how do we use a micropipettor to transfer small volumes.
Gilson or thin micropipettors are one of the most commonly used instruments in a molecular biology laboratory. They're used in conjunction with plastic disposable pipette tips to measure or transfer small amounts of liquids, usually from 0.2 microliters to 1,000 microliters or 1 milliliter. Different pipettes work across different ranges of volumes and are named by the maximum volume they pipette. The different pipettes look very similar, so they're labelled on the top of each push button with a capital P followed by a number which indicates the maximum volume for that particular pipette. A P2 pipettes between 0.2 and 2 microliters. A P20 pipettes between 2 and 20 microliters. A P200 pipettes between 20 and 200 microliters. And a P1000 pipettes between 100 and 1000 microliters. A pipette is an expensive precision instrument. Improper use will damage the pipette, so correct use is essential. You should never use a pipette to measure volumes outside of its range. For example, using a P200 to pipette to lower volumes such as 10 microliters or to higher volumes such as 250 microliters will result in inaccurate volume measurements and will damage the internal mechanism of the pipette. Each pipette has a vertical row of 3 numbers visible in the body of the instrument. To set the volume, you can use either the thumb wheel or, on newer models, the push button. This will cause the 3 number dials in the body of the pipette to rotate and change the volume of liquid that will be taken up. However, the dial numbers indicate different volumes depending on which pipette is used. For example, these 3 pipettes have been set to read the same numbers on the dial, 152. For the P2, this means that the pipette is set to a volume of 1.52 microliters. The bottom 2 dials are in red, indicating 5/10 and 2/100 of a microliter. For the P20, this means that the pipette is set to a volume of 15.2 microliters. The red bottom dial shows 2/10 of a microliter. With the P200, the same visual setting means that the pipette is set to a volume of 152 microliters. This P1000 is set to 0.52, which indicates a volume of 520 microliters. A setting of 100 indicates the maximum volume for this pipette, which is 1,000 microliters. The uppermost number is in red, which for this pipette indicates the number of milliliters. Once you've set the volume you need, attach the appropriate pipette tip to the end of the pipette. P20 and P200 pipettes use the same yellow tips. P1000 pipettes use larger blue tips. Another type of tip called a filter tip is often used in the laboratory. The sterile filter within each tip can help to prevent contamination. Hold the pipette with one hand with the narrow side resting in the palm of the hand. Add the pipette tip by gently but firmly pushing the pipette into the pipette tip, which is held in a tip box. You may need a little practice to learn to apply the right amount of pressure to give a good airtight seal between the tip and the pipette. Liquid is drawn in and expelled using the pipette's push button. Gently apply pressure to the button with your thumb until you feel a natural stop. This is called the first stop. The distance you need to push the push button down will vary depending on the volume you require. A P20 set to 2 microliters will require less push button movement than a P20 set to 20 microliters. So to use the pipette, push the button down to the first stop. Then, keeping the push button at this level, place the pipette tip about 2 millimeters into the liquid you wish to draw up. Release the push button by slowly allowing it to return to its original position. Pause for a second to make sure all the required volume of liquid has been taken up into the tip. This is especially important when using more viscous liquids which take longer to draw up. Withdraw the pipette tip from the liquid and place it inside the recipient container. Slowly push down on the push button. This will release the liquid in the pipette tip into the tube. This time push beyond the first stop. This ensures that any residual liquid is expelled from the pipette tip. Fully withdraw the pipette tip from the liquid before you release the push button. So to recap, push down the push button until you feel some resistance. Place the pipette into the liquid and slowly release the push button. Release the liquid from the pipette by pushing past the first stop to fully expel the liquid from the pipette tip into the recipient container. Withdraw the pipette from the container before you release the button. For very small volumes, the aspirated liquid can hang in a drop from the end of the tip and may not be transferred to the tube at all. So you should touch the tip to the inside wall of the recipient container whilst expelling the liquid. Once you've aspirated the liquid, release the pipette tip from the pipette by pressing down on the tip ejector. This is the small white button at the top of the main body of the pipette. Be sure that the attached pipette tip is inside the appropriate waste container before pressing the tip ejector. A new clean pipette tip should be used with each new liquid, or if the tip touches any surface or any liquid other than the one you're pipetting. If in doubt, change the tip. Do not use a pipette without a tip attached. Liquid should never enter the body of the pipette. It will cause the pipette to corrode and will be a major source of contamination between liquids and experiments. Do not use a pipette past its volume limits. This causes pipetting inaccuracies and also damages the pipette. If you're having trouble attaching a tip to your pipette, don't repeatedly jam the pipette into a tip. This can damage the pipette. And if the tip doesn't stay on the end of the pipette, simply repeat the procedure with a new tip. When pushing down the push button to take up a set volume of liquid, don't push past the first stop. If you push past the first stop, the volume you then take up will be too large. When taking up liquids, don't simply let go of the push button. The liquid can be sprayed around the inside walls of the tip and up into the pipette body, causing inaccurate volume dispensing and pipette contamination. Make sure you release the push button in a controlled manner. If you don't pause after you release the push button, there won't be enough time for the correct volume to be taken into the tip. Air will be taken in instead. Also, when you're withdrawing a large volume from a narrow container, make sure the tip stays below the surface. Whenever you have liquid in a pipette tip, don't lay the pipette down. Liquid can get into the body of the pipette, which causes cross-contamination, pipette damage and inaccurate pipetting. And finally, remember when pipetting small volumes, touch the pipette tip to the side of the tube to ensure the liquid is released into the recipient container.
So let's discuss now, how do we read a micropipettor? So whenever you see a P and then a number, P1000 for example, the 1000 is referring to the fact that the maximum volume that it can dispense is 1,000 microliters. Now, in terms of the minimum volume that pipette can be used, there's a 10% rule. So if the maximum is 1,000, the minimum is going to be 10%, which is going to be 100 microliters. So a P1000 is going to be used to dispense a volume between 100 microliters and 1,000 microliters. A P200, 200 means that the maximum is 200 and the minimum is 10% of that, which would be 20. So a P200 dispenses 20 microliters to 200 microliters. A P20 can dispense a maximum of 20 microliters, minimum of 10% of that. So that would dispense volumes between 2 to 20 microliters. There are smaller pipettes. We don't use them in micro, but in molecular biology, we also have what's called a P2, which dispenses 2 microliters is the maximum, 0.2 microliters at the lowest end, the minimum. But those are not routinely used for microbiology. But a P1000, a P200, and a P20 could be used. Now, when we read a pipette, what we have to think about is that if we look at the dial, so here's the dial, for example, and the number at the top, you always want to think of that's the maximum placeholder. Maximum placeholder. So a P1000, the maximum placeholder is the 1000th position. So 100 reads 1 in the thousands, 0 in the hundreds, 0 in the tens. So that is set to a volume of 1,000 microliters. So that is the maximum that the P1000 should go to. Notice the next one to the right, 050. There is a 0 in the 1000th placeholder, because remember that the highest it goes is 1,000. So there's a 0 in the 1,000 placeholder, a 5 in the 100, 0 in the 10, and nothing in the ones. So that is set to 500 microliters, right? It's not 1,000, so 500 microliters. If we look at the next one down, it has 0, 2, 8, and then a little dash. So 282. Each of those little lines is 2 microliters. So this pipette is set to a volume of 282 microliters. So this is how you read a pipette. Now, what you need to think about is how do we convert between milliliters and microliters? Because remember that in the last diagram, when you do your pipetting, you're transferring 0.1 milliliters, right? 0.1 milliliters. That needs to be converted to microliters because our micropipettors are set in microliters. So our conversion is 1 milliliter is equal to 1,000 microliters. So if I want to convert 0.1 milliliters to microliters, what I need to do is I need to take 0.1 milliliters. And we're going to cross cancel. The units I already have need to go on the bottom. The units I'm trying to convert to go on the top because I'm trying to convert from milliliters to microliters. So now I need to plug in my numbers. 1 milliliter is equal to 1,000 microliters, right? That's my conversion. 1 milliliter is equal to 1,000 microliters. So now when you're doing this math, you do not need a calculator. You will not be allowed to use a calculator. This is math that you should be able to do in your head or on a scratch paper without using a calculator, because all it is moving the decimal point. It's very, very simple math. So if I take 0.1 and I times 1,000, so if I have 1,000 groups of that, should that number get larger or smaller? So should the 0.1 get larger or smaller if I have 1,000 groups of that? The answer is it's going to get larger, right? Because if I have 1,000 groups of that, my number is going to get larger. So now what you need to think about is which direction is moving the decimal point going to make the number larger? Is it going to make it larger if I move the decimal point to the right or to the left? So let me write this out down here so we have a little more space, 0.1, and we're going to multiply by 1,000. So if I multiply by 1,000, we said that it gets larger, right? The number is going to get bigger. And if we're multiplying by 1,000, notice there's 3 zeros. So that means that I need to move my decimal point 3 places. Now, 3 places to the right or 3 places to the left? The answer is 3 places to the right because that's going to make the number larger. So we go 1, 2, 3. The decimal point would be here. This gets a loop, gets a 0. This gets a 0. So that is equal to 100 microliters. So when we're transferring 0.1 milliliters, we are transferring 100 microliters. So if I were looking at a pipette and I have my 3 spots, I would have a 0 here in the thousands. I would have a 1 in the hundreds. So here's my 1. I would have a 0 in the tens, and I would have a 0 in the ones. That's what my pipette would be set at if I'm pipetting 0.1 milliliters or if I'm pipetting 100 microliters. So 0, 1, 0 would be pipetting 100 microliters. That is also the minimum for that pipette. A P200. P200, the maximum is going to be the hundreds placeholder. So this top number is hundreds. Then we have tens and ones, and then decimal point after that. So 200 is the same as saying 200 microliters. That's the maximum. Now, if it reads 050, we have 0 in the hundreds, 5 in the tens, nothing in the ones. So that pipette would be set to 50 microliters. Then we have our next pipette. It reads 158 and then a little dash to the left of that. So that's 2. So 158.2, because again, the top placeholder is the hundreds. So 158.2 microliters. If we're looking at a P20, the maximum is going to be 20 microliters. So the number at the top is no longer the hundreds. The number at the top is now the tens, and then the ones, and then the tenths, and the hundredth. So meaning the decimal point is after, so starting where the red number is. So if it reads 200, that is going to be the maximum, which is the 20 microliters. So notice if you look at the P200, when the P200 reads 200, that's the maximum volume, which is 200 microliters. If you're looking at a P20 though, and it reads 200, that is not 200, that is 20. So when you're reading your pipettes, you have to make sure that you're aware, is it the P200? Is it the P20? Is it a P1000? Because that definitely makes a big difference in the volume that's being pipetted. If it reads 050, 0 in the tens, 5 in the ones, and then nothing after the decimal, so that pipette would be set at 5 microliters. And then lastly, if it reads 158 and then one little line to the left, that is going to be 15.82 microliters. And so this is how we read our micropipettors to determine our volumes. So again, if we're looking at a P20, for example, the maximum is going to be 20. The minimum is 10% of that, which is 2. So when we're working with a micropipettor, we never want to turn the dial. There's this little area here, this little wheel that turns. We don't want to make our numbers in the little dial there to read outside of that. So it should never read above 200, and it should never read below 020. 2 microliters is the minimum. So if we're looking at a P20 and it reads 050, nothing in the tens, 5 in the ones, nothing after the decimal point, that pipette is set to a volume of 5 microliters. So if we are reading the micropipettors, the P20, 050, we just said is 5 microliters. If that is a P200 and it reads 050, remember that the top placeholder is now hundreds. The 5 is now in the tens, nothing in the ones, so that reads 50 microliters. If we are reading that in a P1000, the top number is going to be the thousands, then the hundreds, then the tens. So if it reads 050, it is now set to 500 microliters. So notice that these dials all look the same, yet the volumes that they're set at would be very different because it's dependent on the maximum. So when working with our micropipettors, there is a push button or a plunger And that push button has two positions, meaning that when I pipette, I can get to what we call the first stop, and the first stop is where I start to feel resistance. But if I push a little bit harder, I can get to what we call the second stop, meaning I can push down further and get to the second stop. Now, the reason that that's important is that when I'm doing my pipetting, the only time I go to the second stop, meaning the only time I'm going to push all the way, is if I am doing what's called the blowout phase, meaning I'm trying to get all the liquid out of the tip. That's the only time that I would go to that point. When I go to draw up liquid, I only push the plunger to the first stop. I would stick my tip in the liquid. I would slowly release my thumb, take my tip out of the liquid, and then transfer it wherever it's going to go. And when I do that to get the liquid out of the tip, that's when I'm going to go to the second stop. That's when I'm going to go to the blowout phase. I go to the second stop when I'm trying to get the liquid back out of the tip. But otherwise, I don't want to push to the second stop. If I push to the second stop when I'm trying to draw up liquid, I'm going to pipette too much. It's not going to be accurate. Now, in terms of other parts of your micropipettor, we have our thumb wheel. That's the little dial that you turn. This little thumb wheel here is what's going to be used to set the volume. This is the volume display on the front. They also have a tip ejector button. So there's this button here, and that's how you get the tip off. Your tip is going to go on the barrel. I will make a video demonstrating using a micropipettor as well as doing the standard plate count. But you already saw in that previous video an example of how to use your micropipettor. So the way that we use our micropipettor is that we have to start by twisting the dial to set the appropriate volume. So again, if I'm trying to pipette 100 microliters and I'm using my P1000, I need to make it read 010 on my micropipettor. So the first thing we have to do is we have to twist the dial to set the volume that is desired. Now, in many pipettes, they also have a lock switch, meaning that there's a function on it that keeps the dial from moving as you're pipetting. Again, you'll see that in the video. You would take your pipette and you would add a disposable pipette tip. So like you saw in the video, you just firmly push into the tip box to pick up the tip. You don't want to jam it repeatedly in the box. That's not necessary. But you also need to make sure that you have some firm pressure. Otherwise, the tip is going to fall right off. So you just gently push into the tip to add the pipette tip to the barrel. Then you're going to push the plunger to the first stop. So again, to that place where you feel that resistance. You're going to take the pipette and you're going to put the tip into the liquid that you're trying to draw up. Now, when I do this, I'm careful that I don't go, especially if I'm using like a large volume, I don't want my tip to go too far down because then I end up with liquid on the outside of the tip. So for me, I try and keep my tip just barely inside the liquid. But you need to also be careful and watch because you need to make sure that your tip is submerged in the liquid at all times when you're trying to draw the liquid in. Because if it's not, if it comes out of the liquid, you're going to draw in air and you're not going to have the appropriate volume. So it's a fine balance. So I have my push button, my plunger, push to the first stop. I put my tip into the liquid that I want to draw up. I slowly release the plunger to retrieve the liquid. Now, I don't want to just let go of my thumb from holding that plunger down because if I do that, it's going to slosh liquid into the barrel of the pipette, which I don't want to do. So I would just slowly release my thumb and the liquid would get drawn up into the tip. Then I would move my pipette tip into my desired tube. So I move my tip into my desired tube and then I need to get the liquid out of the tip. So at this point, I push to the first stop and then the second stop. So that's my blowout phase. So this is when I push to the second stop to get all the liquid out of the tip. Once I get the liquid out of the tip, I keep my thumb on the plunger. I remove the tip from the liquid, and then I release my thumb. If I still have my tip in the liquid and I push all the way to the second stop and I'm still in the liquid and I release my thumb, it's going to draw the liquid right back out. So it's really important that you push to the second stop, you get all the liquid out of the tip, take your pipette out of the liquid, and then release your thumb. And then you can inject your tip into a tip waste container. So what we would do if we were in the lab is that we would have you practice doing your pipetting. We would want to make sure that you can accurately pipette in order to do your standard plate count. And so what we would give you is we would give you a little sheet. It's a laminated paper, and we would have you practice pipetting these various volumes. So you would add 150 microliters to student number 1. That would be you. Then you would change the dial and you would add 300 microliters to the next circle, and then 600 microliters. We would tell you to cross out the 800. That volume is too big. It spreads out too much, so we wouldn't have you do that volume. Student number 2 would do the same thing. They would pipette 150 microliters, 300 microliters, 600 microliters. And the reason we do that is we would have you compare. We use a green practice die, and we would have you compare the size of your green puddle of liquid. If the sizes are similar, that means that you and your partner likely are pipetting the same volume. However, if you're both doing this and you're noticing that your partner's, green die is much larger, the circle is much larger than yours, somebody is not pipetting accurately. And we would want to figure out where the problem was happening, meaning why are we not getting accurate pipetting? And so this would be the first thing we would do is we would practice pipetting first to make sure that our pipetting could be accurate so that our standard plate count would also be accurate. So when we do our standard plate count, we would have you get the following items per pair. So we have our 4 nutrient agar deeps. Notice it says hold in a 55-degree water bath. So we don't want to take these agar deeps out of the 55-degree water bath until we're ready to go. If we take them out at the beginning of the experiment and we set them on our desk, that is going to be problematic because by the time we're ready to use them, they would solidify in the tube. So we get the rest of our experiment set up first, and once our cereal dilutions are made and we're ready to actually pour our plates, then we would take our nutrient agar deeps out one at a time. We would need 4 sterile Petri plates, right? We need 4 sterile Petri plates. They don't have any agar in them yet because again, you're going to use nutrient agar deeps to pour into the plate. You would use a P1000 micropipettor. So you and your partner would have one P1000. You would have a P1000 micropipette tip box, meaning it contains the tips for the P1000. You would have a plastic beaker to put used tips in. We call it a tip waste beaker. You would have a 100 milliliter beaker for micropipette practice, meaning it would have the dye to practice with. You would need sterile deionized water blanks, 3 of them, right? You need 3 tubes that have 9.9 milliliters of sterile deionized water. You would need 1 E. coli stock culture. That's the culture you're trying to determine the titer. And then you would need 1 test tube rack for these large tubes. These large tubes would not fit in those other racks that are normally at your desk. And so we would have you gather all of these items before you begin your standard plate count. Now, what you would do in this experiment is that you would need to start by making your cereal dilutions. right? And so you would take your P1000 and you would set it to 100 microliters. So we have our P1000, we would set it to 100 microliters, and we would vortex to mix the stock culture. So we would vortex, we would mix that stock culture, and then we would use our micropipettor to transfer 100 microliters into a blank that contains 9.9 milliliters of water. Remember that that's going to make the dilution in that tube 10 to the negative 2. So when we start, before we add anything to these sterile water blanks, we would label these on the glass, the water blanks. We would label 1, 10 to the negative 2. 1 we would label 10 to the negative 4. And the last one we would label 10 to the negative 6, so that we keep track of our dilutions. So we add our 100 microliters into our water blank. And then once we add that bacteria, we would need to mix well. We would need to vortex before we take liquid from this tube and transfer to the next tube. We have to mix well so that we make sure and the bacteria is evenly distributed so that our accuracy is done well. So we have to mix well before we take our pipettor and pick up 100 microliters again. And then we would transfer the 100 microliters to this tube, and we would mix well, so vortex to mix. After we vortex to mix, pick up 0.1 milliliters, transfer it to another water blank. So we would start our experiment by performing our serial dilution. We would make our 3 tubes that have our 10 to the negative 2, 10 to the negative 4, and 10 to the negative 6. Now, our 10 to the negative 2, we're not going to use for anything else. That one was just made to make my 10 to the negative 4. But once I make all 3 of my serial dilutions, I can put my 10 to the negative 2 aside because I don't need that anymore. Now, after I make my serial dilutions, then what I would do is I would get out my 4 sterile Petri plates and I would label my Petri dishes. And I would make sure to label them on the bottom side, so not the lid, the bottom side of the plate. And I would label them with my initials. I would label them with the date. And I would also label them with my final dilution factor that's on my plate. So that would be my 10 to the negative 4, my 10 to the negative 5, 10 to the negative 6, and 10 to the negative 7. So I would label all 4 plates. And when I do this, I would set up my plates around my flame. Because remember that when my Bunsen burner is going, my sterile field is around the flame. So I would set up these plates so that the lid part is on the top, meaning the base is on the bottom, because you're going to pour the agar into the bottom part of the plate. You don't want to pour into the lid. So we would pour into the bottom part. So we would have to set up our plates around the flame and set them up. I would set them up in order: 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. So I would label my tubes, or label my plates. Then once I have my serial dilutions, I can take out 1 nutrient agar deep at a time. So I would go to the water bath and I would pick up a nutrient agar deep, and that nutrient agar deep is kept at 55 to keep it liquid. So I would get my nutrient agar deep and I would bring it to my station, and I would add 1 milliliter, which is 1,000 microliters, from my 10 to the negative 4 plate. I would add that to the agar deep. So I add my bacteria, I mix, and then I pour it into the plate. So that when I pour it into the plate, my bacteria is going to be mixed with the agar. And when it solidifies, the bacteria is going to be within the agar. And so we're looking for a plate that has a countable number of bacteria. So when we pour these plates, once we pour the agar into the plate, we do not want to move our plates. If we try to move our plate after that, it's going to slosh the agar around and it's going to contaminate your desk because it has bacteria in it. And it's also going to end up with agar on the lid instead of just in the bottom of the plate. And your count is not going to be accurate. So you want to make sure that when you pour these, you pour them close to the flame and you pour them in a place where you're not going to be worried about having to move them after. So I pour this liquid into a plate and I let it sit and give it time to solidify. Then I would go grab another nutrient agar deep, and now I would use my 10 to the negative 4 plate, and I would add 0.1 milliliters to another agar deep, and then pour that into my plate that's 10 to the negative 5. I would then go and get another nutrient agar deep. I would start with my 10 to the negative 6 dilution this time, transfer 1 milliliter, pour it into a plate. Grab another agar deep, transfer 0.1 milliliters from my 10 to the negative 6 into the agar deep. Once the bacteria is added to this tube, I pour it. And then again, I let all 4 plates sit until they solidify. And then I take my 4 plates, I would rubber band them together. I would make a stack of 4 plates. And then I would turn them agar side up, because remember, we always put our plates agar side up. So we put them agar side up and we put them in the incubator. And we would let them grow and then look at our plates later to determine our titer. And so this is how we would do our standard plate count. There will be a video demonstrating how this is done so that you can kind of see it in action. It makes it a lot easier to see when the experiment is done. But this part is just simply kind of walking you through the steps. So some tips for when we do our standard plate count. Number 1, only open the tip box as needed. We don't want to keep the lid off the tip box for long periods of time because the tips are sterile. They have been autoclaved, meaning they've run through our autoclave, which is a giant pressure cooker essentially, which makes the tip sterile. If we leave the lid open for long periods of time, those tips are no longer sterile because things from the air could fall in. So when we do this, we make sure that our tips are only open as long as we need them, and then we close the lid right away. You always want to make sure to vortex thoroughly before going into any tube. So anytime you're going to transfer liquid from one tube to another, you need to mix well in order to get it mixed well to transfer to the next tube. Just like in our aseptic technique, you need to make sure to flame the opening of the tube when you go into the tube and before you put the cap back on, right? So before and after. If you must set the cap down, which again, hopefully you don't because you could put the cap between your pinky and ring finger, but if you absolutely must set the cap down, make sure to place the cap with the opening face down so that, again, bacteria doesn't fall into the lid. You would use a new pipette tip for each dilution. Yes, you are transferring the same bacteria from one tube to the next. However, it's a better idea to get a new tip because sometimes you'll have residual liquid in the tip, which then might affect your pipetting accuracy. So it's a good idea to get a new tip for each dilution. Never hold your pipette upside down, meaning if I have liquid in my pipette, I don't want to turn it upside down so that the liquid is no longer in the bottom, the liquid is on the top, because the liquid then is going to go into the barrel of the pipette and it's going to contaminate it. So you always want to keep your pipette oriented so that the tip and the liquid are facing the bottom, and you never turn it over. You never want to set that pipette with liquid in it on your desk. Only take the agar deeps as needed. So again, you don't want to grab all 4 agar deeps at the beginning because they will solidify before you get a chance to pour them. So you only grab them as you need them. Make sure to pour agar into the plate and not the lid. The lid is less shallow. It's not as deep. So you want to make sure that you don't pour the liquid into the lid, but instead that you pour it into the base of the plate. And then lastly, once the plates are poured, do not move them. Again, if you try to move them once you've already poured them, the liquid is going to slosh around and it's either going to contaminate the desk, it's going to end up with bacteria in the lid, or your count is going to be inaccurate. And so it's important that you make sure and don't move your plates once they're poured. So now we're going to go through day 2 and talk about the readout. So on day 1, we would set up our experiment, perform our serial dilutions, and then plate them onto plates. And so when we would come into the next class period, we would do our readout to determine our titer. And so we're going to talk about how we do our calculations. So just a reminder about our serial dilutions and our dilution factors. Remember that the way that we calculate our dilution factor is that we take the volume that we're transferring. So starting at the top, we take 0.1 milliliters and we transfer that into a final volume of 10 milliliters. So we have 0.1 milliliters into 10 milliliters. Now we want to convert that into 1 over some number. So I had to move my decimal point 1 place to the right on the top, which means I had to move it 1 place to the right on the bottom. So now my dilution would be 1 over 100. 1 over 100 would be the same as 1 over 10 to the second. So 2 is the exponent because there's 2 zeros in 100. 1 over 10 to the negative 2 is the same thing as saying 10 to the negative 2. So my dilution factor, the amount that I'm diluting each time, is 10 to the negative 2. Since I start with my stock and I dilute it 10 to the negative 2, my final dilution factor in the first tube is going to be 10 to the negative 2. That's the dilution in that tube. That's why we call that the final dilution factor. Then we dilute it 10 to the negative 2 more. So we transfer 0.1 milliliters from that one and add it to 9.9 milliliters of a water blank. My dilution factor is still 10 to the negative 2. That's the amount we're diluting. But my final dilution factor in the next tube is going to be my 10 to the negative 4. I simply add my exponents because I start with 10 to the negative 2 and I dilute it 10 to the negative 2 more. So my final dilution factor in that tube is 10 to the negative 4. Then I take from my 10 to the negative 4 and I dilute that again 10 to the negative 2. And my final dilution factor is going to be 10 to the negative 6. So that is going to be my calculations for my final dilution factors in my serial dilutions. Now I take those serial dilutions and then I use those to plate onto a plate. So I take my 10 to the negative 4 plate and I transfer 1 milliliter. If I transfer 1 milliliter, my final dilution factor on that plate is 10 to the negative 4. Because remember, my titer is going to be the number of bacteria per milliliter. So if I transfer 1, my final dilution is still 10 to the negative 4. Now, if I take from my 10 to the negative 4 plate and I transfer 0.1 milliliters, notice that's 10-fold less. 0.1 is 10-fold less relative to 1. So 10-fold less means that my exponent changes by 1 number. So now it's going to be 10 to the negative 5. This, right, 10 to the negative 5 is more dilute than 10 to the negative 4. I'm transferring 10-fold less. Then I take from my 10 to the negative 6 plate, right, and I transfer 1 milliliter. I end up with my final dilution on that plate, 10 to the negative 6. The last and final plate, I take from my 10 to the negative 6 and I transfer 0.1 milliliters. So that's 10-fold less than 10 to the negative 6, which gives me 10 to the negative 7. So now, if you think about this, I have my 4 plates. My plates are labeled 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. Which plate would you expect to see the most bacteria on? And the answer is you would expect to see the most number of bacterial colonies on your 10 to the negative 4 because that one is the most concentrated, meaning it's the less dilute. So what you expect when you look at these plates is your 10 to the negative 4 is going to have the most colonies. 10 to the negative 7 is going to have the least. And so you should see that trend, and we'll look at an example of that in a minute. So what I want you to do is I'm giving you a different example for a serial dilution. And what I want you to do before the Zoom meeting is I want you to look at this diagram and I want you to calculate what is the dilution factor, meaning how much are we diluting in our serial dilution. I want you to calculate your final dilution factor of each tube. So let's say we call this tube A, tube B, and tube C. So I want you to tell me what is your final dilution factor in tubes A, B, and C. And then also, what is the final dilution factor for each plate? So if I take from tube B and I transfer 1 milliliter, what would my final dilution be on this plate? Or if I take from tube B and I transfer 0.1, what would my dilution be on that plate? If I take from tube B and I transfer 0.01, what would my final dilution be on that plate? So this is a chance for you to get some practice and make sure that you understand how to calculate your serial dilutions. How do you calculate your dilution factors? How do you calculate your final dilution factors? So I want you to do this before our Zoom meeting, and then I will put you in a group and you will discuss your results with your group and see if you came up with the same answers. And so practice your calculations. Now let's talk about how we analyze our results. So what we're looking for in our experiment is we are trying to determine our titer. And our titer is the number of bacteria per milliliter. So what we do is we look at the number of colonies that are on the plate. And the number of colonies that are on the plate is referred to as CFU, which stands for colony forming units. Colony forming units is your calculation or your count of how many colonies are on the plate, right? And so you're going to look at how many colonies you have. Now, remember, each colony, each little mound of bacteria, was originally one bacterial cell that reproduced asexually. It simply made a copy of itself. So that one cell went from 1 to 2, and then from 2 to 4, 4 to 8, to 16, 32, 64, and so on and so forth. And so each colony represents one bacterial cell that was on the plate initially. So when you count your number of colonies, that tells you the number of bacteria that were on that plate. Now, what plate do we use to count? Well, we need to have the plate that has a countable number. So notice that we poured 4 plates. The reason that we poured 4 plates is that you're going to have a range of colonies. Some plates are going to have way too many colonies to count. There's going to be no way that you can accurately do that calculation. There will be other plates that will have too few colonies to count, meaning there aren't enough for that count to be accurate. So when we look at our plate, if it has less than 30 colonies, we say that that plate has too few colonies to count. So if we have less than 30, too few colonies to count, it's not going to be an accurate representation. If we have more than 300, we have too many colonies to count. There's no way that we could accurately count that many colonies. So what you're looking for is, remember that you have your 4 dilutions, and your 4 dilutions are going to have different numbers of bacteria. So let's say on my 10 to the negative 6 plate, let's say that we have 82 colonies, 82 colony forming units, right? That would be my plate that has between 30 and 300. Now let's talk about, should you have more than one plate that has between 30 and 300? And the answer is theoretically not. Notice that between 30 and 300, that's a 10-fold difference. Each of those serial dilutions is a 10-fold change. So you shouldn't have 2 plates that fall within that range. So for example, let's say that my 10 to the negative 6 has 82 colonies on it. My 10 to the negative 7, we would expect would have tenfold less. So maybe it has 8 colonies on it. That would be too few to count. And let's say that if we have 82 on that plate, my 10 to the negative 5 maybe is going to have 810. So meaning it's not exactly a tenfold change, but approximately. So 82 colonies on my 10 to the negative 6. On my 10 to the negative 5, it's going to have 810 colonies. So my 10 to the negative 5 would have 810 colonies. My 10 to the negative 4 is going to have 10 times that amount. So maybe it has 8,200, right? Approximately tenfold more than the number of colonies on my 10 to the negative 5. So notice that if there's a tenfold change approximately for each of those plates, you should only have one plate that falls between 30 and 300. So in the example I gave here, I would count, right? I would use my plate that has my 82 colonies because that's the plate that has between 30 and 300. So when I calculate my titer is going to be my original cell density, right? My original cell density, which we abbreviate OCD. The way we calculate that is we take our CFU, so our colony forming units, the number of colonies on the plate, and divide by the final dilution factor that's on that plate. So let's do an example. So my calculation would be 82 CFU, colony forming units, divided by 10 to the negative 6 milliliters. That's the dilution that's on that plate. So I have 82 colonies on my plate. That is 10 to the negative 6. So if I take 82 and I divide by 10 to the negative 6, it's the same thing as saying 82 times 10 to the positive 6. So 82 times 10 to the positive 6. Now that is still not in proper scientific notation because you want that number to be between 1 and 10. So if I have 82 times 10 to the 6th and I need to make that between 1 and 10, I need to take my decimal point and I need to move it one place. So equals 8.2 times 10 to the 7th. It was 10 to the negative or 10 to the 6th. When I move it one place, it's now 10 to the 7th. And then my units would be CFUs per milliliter. Now, if you're struggling with what to make this exponent when you convert it from 82 to 8.2, what I would recommend doing is, draw it out and then work backwards. So let me show you what I mean by that. So 82 times 10 to the 6th, that would be the same as, there's the decimal point. Move it 6 places. 1, 2, 3, 4, 5, 6. Each of these gets a loop. So that number is going to be 82 million. Now, if I need to make it 8.2, notice what has to happen. So if I make it 8.2, my decimal point is going to be 1, 2, 3, 4, 5, 6, 7. So that's why it's 8.2 times 10 to the 7 CFUs per milliliter. That would be my original cell density. So that means that in that stock culture that I started with, that titer, that concentration is 8.2 times 10 to the 7 CFUs per milliliter. Now, notice that my titer is CFUs per milliliter. There's a question in your question set that says, how do you calculate the total number of microorganisms? So what that means is if I only have 1 milliliter, right? If my original stock was only 1 milliliter, my total number of organisms would be 8.2 times 10 to the 7th, because that's the amount of bacteria per milliliter. So if I only have 1 milliliter, that is going to be my total number of bacteria. But what if I have 2 milliliters? So if I have 2 milliliters, right, so if my concentration is 8.2 times 10 to the 7th CFUs per milliliter, and I want to know the total number of bacteria in the tube, if I have 2 milliliters, I need to multiply by 2 so that we can cross-cancel our milliliters. So that would give me my total number of bacteria. So when I multiply by 2, I take my 8.2, multiply by 2, I get 16.4 times 10 to the 7th. That is not in proper scientific notation, so I need to move the decimal point one place. So that would be 1.64 times 10 to the 8th. That would be the total number of microorganisms in the stock if there were 2 milliliters when we began. So our titer is the number of bacteria per milliliter. If we want to determine our total number of microorganisms, we need to take our titer times the number of milliliters. That is going to tell us the total number of microbes that was in that stock solution. And so this is how you're going to do your calculations. And so what I'm going to do is on the next slide, I'm going to show you an example of a result that you might get with your standard plate count. And what you're going to have to do is you're going to have to calculate your number or your original cell density. You're going to have to determine the titer. So let's take a look. So this would be an example of what this would look like. So let's say that the first plate, that's your 10 to the negative 4 plate. And on that plate, it has 6,467 colonies. And then on your 10 to the negative 5 plate, it has 594 colonies. And then on your 10 to the negative 6, it has 62 colonies. And on your 10 to the negative 7, it has 5 colonies. So you need to think about, if you look at these 4 plates, you only use one plate to determine your titer. You're only going to use one plate. So you have to think about which of those plates should you use and why. And then use that plate to calculate your original cell density. Now, one of the things that I want you to notice, and you can see this even more abundant or more prevalent in the lower concentrations, notice how some of these colonies are very large and some of these colonies are really small. And so you might wonder, well, do I need to include counting the small colonies or just the big ones? The answer is that all of the colonies count. Any of those little dots would count as a bacteria. So each of these little dots is a bacterial colony, which means that that was one bacteria that divided asexually and gave rise to 2 and then 4 and then 8, etc. So why then do we have some colonies that are quite large versus some colonies that are small? And the answer is that, remember, we take our bacteria and we mix it with the agar, and then we pour that into a plate. So what ends up happening is you're going to have some bacteria that's on the top of the agar, meaning that it's interacting with the oxygen in the atmosphere. And you're going to have some bacteria that's embedded in the agar, because again, the bacteria was mixed with the agar. And so you're going to have some bacteria that are within the agar, meaning they're not on the top, but they're actually embedded in the agar. That is going to affect how fast that bacteria replicate. And the reason is, and what you're going to learn throughout the semester, is that when bacteria grow aerobically, meaning in the presence of oxygen, when they grow aerobically, they get the greatest yield of ATP. So doing aerobic cellular respiration, they can get between 30 to 32 ATP per glucose. The bacteria that are growing within the agar, in the middle, they are growing anaerobically, meaning without oxygen. And if they're growing anaerobically, and instead of doing aerobic respiration, they do fermentation, when they do fermentation, they only get 2 ATP per glucose. So notice the energy yield is very different if bacteria grow with oxygen or without. So with oxygen, bacteria are going to grow much faster. So those large colonies are the bacteria that are on the top of the agar, meaning they're interacting with the oxygen in the atmosphere. The teeny tiny little colonies, Those are the bacteria that are embedded in the agar. They're growing anaerobically, they're growing without oxygen, and they're growing more slowly because without oxygen, they don't have as high of an ATP yield. So they grow a lot slower because they don't get as much energy out. And so that's why you're going to see these differences in colony size. That doesn't mean that you can exclude one or the other, because any colony that shows up on the plate is clearly a bacteria that was on the plate. So that's why we have to count all of our colonies. All of the colonies count, no matter what the size of the colony is, if they're large colonies or small colonies, etc. They all count. Now, let's say that we had a plate that basically, you weren't sure about the number of bacteria. Let's say it's our 10 to the negative 5 plate. Notice that on that plate, to count every colony that's on that plate would be really difficult because there's so many. So one of the things that we could do is actually divide our plate into quadrants. Divide it up. So I might divide my plate into 4 quadrants. Now, don't mind my non-straight lines, but what you would want to do is then you would count one of the quadrants, and you would count that quadrant for the number of bacteria. You would want to make sure that you don't pick the quadrant that appears to have the most. You don't pick the quadrant that has the least. You want to pick a quadrant that is representative, meaning that it has a number somewhere in between. Because if I count the quadrant with too little bacteria, meaning the one with the least, my estimate is going to be too low. If I pick the quadrant that has the most, my estimate is going to be too high. So what do I mean by that? So ignore the 594 for a minute. Let's say that we counted a quadrant, and within that quadrant, there were 50. So again, ignore the 594. I'm just doing an easy number for math. So let's say that we counted one of our quadrants where my plate was divided into fourths, and that quadrant had 50 colonies on it. I would not record my colony forming units as being 50. There are 50 colonies on 1/4 of the plate. So my total number of colonies on the plate is you would assume that there's 50 in this quadrant, 50 in this quadrant, 50 in this quadrant, 50 in this quadrant. So 50 times 4, you would record that the total number of bacteria on that plate is actually 200, right? 50, 100, 150, 200. So that's how many colonies you would record on the plate. Now, for the purpose of this, we already counted the colonies for you, so you don't have to sit there and count them. But if you were doing this in lab, I just wanted you to be aware of how would you count these colonies. And when you're counting them, if I was doing this in the lab, I would use a permanent marker on the bottom side of the plate, so not on the agar itself. I'm not going to open the plate and touch the agar. But on the bottom side of the plate, what I would do is each time I count a colony, I would use a permanent marker and I would mark that colony so that I know that I've already counted that. And that way you keep track of which colonies have already been counted. So that's just to give you an idea of how you would count these colonies. But instead, we already gave you the numbers. So you're just going to use this diagram and you're going to calculate your original cell density. Make sure that you write the appropriate units that you would have for your original cell density. And then we will discuss our results in our Zoom meeting. And we'll see if we got our correct titer, if you calculated the correct original cell density. And so this concludes our standard plate count.